video, we will be talking about the transcription process from DNA to RNA. DNA is a double-stranded molecule that forms a double helix shape. Since DNA cannot leave the nucleus or the cell, messenger RNA needs to be created to carry instructions for protein synthesis to the cytoplasm. One strand of DNA is used as the template. Then it is matched up with complementary RNA nucleotides, which are the same as DNA nucleotides, except adenine goes with uracil instead of thymine. DNA has an anti-parallel structure, which means that even though the strands are parallel, they are oriented in opposite directions. The strand on top 5' end, 3' end, while the bottom strand goes from 3' to 5'. The enzyme that synthesizes mRNA from a DNA template is called RNA polymerase. It can bind to a strand of DNA at a site called a promoter right before the regulatory sequence. An example of a promoter would be the TA box, which is named for its pattern of base pairs. As RNA polymerase moves down a strand of DNA, the strand unwinds. One strand will act as a template, while the other is known as the coding strand. The pre-mRNA that is sent from the template strand has almost exactly the same microgenous bases as the coding strand, except instead of thymine, it's uracil. The RNA polymerase accomplishes this by matching each nucleotide on the template strand with its complementary RNA nucleotide.
Remember that all of this happens inside the nucleus since the DNA cannot leave.
RNA polymerase stops creating pre-mRNA at a point called a terminator. It is there that the enzyme stops moving along the strand and the DNA is rewound back into a double helix shape. As a review, the creation of pre-mRNA has three parts. The first part, in blue, is the promoter, combined template strand. The second part, in yellow, is the transcribed region. This is the part that gets matched with RNA nucleotides to create mRNA. The final part that involves RNA polymerase is the terminator, or termination point. There, the enzyme detects that it no longer needs to replicate the genetic information and can disconnect from the terminate strand. Notice that the pre-mRNA has regions that are highlighted and regions that are not. The yellow regions are exons. Exons are the parts of pre-mRNA that contain the genetic material that codes for protein. The other parts, the one that are not highlighted, are known as introns. Introns are one of the part of pre-mRNA that need to be spliced out. They're known as the non-coding regions, since they're not used in protein synthesis. Once the introns are taken out, you're left with a strand of messenger RNA. Notice the weird endings. On the 5' sign, there's a modified guanine called a 5' count, which both protects the mRNA and helps it adhere to ribosomes for translation. On the 3' end, there is a chain of multiple adenines. This is called a 3' poly A tail, and it protects the messenger RNA strands from degrading while it travels through the cytoplasm. Now that the mRNA contains critical genetic information for protein synthesis and it's fully assembled, it's ready to be sent out to the cytoplasm to create proteins.